Thank you for having me here. It's really exciting. I actually thank you, Svenja, for noticing my title in the OSM. I didn't know too much about JETZON, so it was really cool to read about it. And I'm happy today to present the work that we've been doing on the metabarcoding analysis of Salp diets. And we're hoping to find more about the trophic relationship in the Mesopelagic food webs. And all this work is possible thanks to all my co-authors, Jennifer Costell, Joel, Peter Wiebe, and Anne Buckland for Woodsall and the University of Alaska in Fairbanks. Okay, so let me see, how do I, there we go, yeah. Okay, so most of you are knowledgeable and experts, probably more than me, about the ocean twilight zone, but I'll go ahead and introduce it a little bit. The Mesopelagic is characterized by high biomass and biodiversity of fishes, crustaceans, and other jellyfish, as you all know. The food web dynamics of this area, it's largely unknown, and the motivation for a lot of this work that we're doing is to investigate a little bit more about the diversity and the taxonomy and the structure of the food web. And as I mentioned while talking to you guys, our lab focuses on this gut content analysis of fish and also salps. So I'm going to focus on salps because I worked with them before and I really like them, and I think they're really interesting, especially in this region of the ocean. So as some of you might know, salps are getting a little bit popular. Finally, they're getting some attention. And well, they gather their food. Oh, let me get this started. Yeah, they gather their food by pumping water through a mesh. So this is a salp over here in the video, and Salpa thompsoni, which you might recognize too. And as I described, they pump water through their body. And inside their body, they have a mesh where they trap all the particles in the water. And this mesh gets transported to that orange blob down here, which is their stomach. And while salps are interesting to us because they adjust really quickly to fluctuating food concentrations by the high filtration of rates that they have, because they swim so much. They are well known for their diel vertical migrations. Because of this mesh, they can filter a wide range of prey size. And they also have a very complex life cycle. So they can alternate between asexual and asexual reproduction. And this is very favorable under bloom conditions where they're taking advantage of favorable environmental conditions. So we are interested in learning what are salps feeding on. Is it always the same diet? And for this, we use the Tree of Life gene, 18S, and we use some regions of this gene. So since we know the gut content of salps can be hard to identify under the microscope, it can be time consuming and costly. There's been some studies with electron microscopy that has made it possible to understand a little bit more. But since we don't have access to that, we decided the metabarcoding was the way to go. And metabarcoding, it's a molecular tool to identify using a genetic marker. And it could be of samples or gut contents in our case. So 18S has multiple regions, and they have hypervariable regions highlighted in green here in the diagram that I'm showing. And B4, it's a hypervariable region of this gene and tends to have a good taxonomic resolution and can resolve the species level in certain taxonomic groups of zooplankton. B9, it's also a hypervariable region of 18S. However, these can resolve to taxonomic groups, but not so accurately to species. So now that I set the background of our work a little bit, I'm going to talk about where we collected the samples. So salps were collected in the slop water of the Northeast United States in 2018. I'm showing the map on your left side. 2018 is on the top and 2018 is on the bottom. In this collection events, we were able to separate three different salp species from the zooplankton collections. And one was Salpa aspera, Salpa fusiformis, and Yacisonaria. So those are the three species, and you can see that all of them have the orange blob that I like so much to analyze. And that's the gut. So we dissected the gut and the tissue from each individual and to try to avoid any contamination from the the predator from the Salp itself. We washed it and then we extracted some DNA. We used this DNA to generate amplicons for the two hypervaluable regions that I mentioned for the 18S, the B4 and B9 region. Okay. Then after we generated those amplicons, we sequenced the amplicons in an Illumina MySeq platform. We carried out the bioinformatic analysis on Modur, which is a well-known pipeline for this kind of analysis. And we used the Silva 132 reference database. We also did some statistical analysis using MATLAB for generating some NNPS and some similarity analysis using the Fathom toolbox. Now we're getting to the results, and I'm going to start talking about relative abundance of prey. So we use the sequence numbers that we got from the sequencing in the Illumina, and we transform it for prey groups and subgroups. So these are the groups that we detected for B4. And so you can see the plots are organized by year. So the upper group of bar graphs are 2018 and the lower bar graphs are 2019. And this is only for the hypervariable region of B4. I'm also keeping the species enclosed by the squares, black squares. So Yassistonaria collected in 2019 is over here, followed by Salpa afra, Salpa fusiformis, and the same for 2019. And over here on the right side, we have all the groups that we detected. So as you can see, 2018 had lower sample sizes, but we were able to detect most of the zooplankton groups or expected groups, plankton groups, and in 2019 as well. However, we weren't able to detect any ciliates or any amphipods. And also, something to point out is that we still were able to detect or capture some of the salp or the predator swamping in the samples, and that's highlighted by black. Those are the salps. And in the purple, the deep purple and the light purple, we have dinoflagellates and syndignales. So those were kind of expected prey for the salps. But also we have other groups like fish and gastropods and siphonophores detected by B4. Now, similar analysis and the similar plot layout for V9. For V9, we actually did detect ciliates and amphipods, which are highlighted. The amphipods are in yellow over here, and the ciliates, and there's light orange. And again, we detected predator DNA as well. So we had some Salp sequence numbers, which was significantly high than other groups, but not similar to what the dinoflagellate signal we were getting. So just wanted to, because that was a lot of plots for everybody to see, and like colors, there are too many colors. So I made this kind of summary plot for this relative abundance of the sequence numbers. And we have 2018 B4 on the top and 2018 B9. So I try to keep it with small numbers. But I just wanted to show that the groups that we were interested in were detected by both markers. And we were seeing similar detection levels, except for those groups that B9 is better at capturing, right? So we get more plankton groups with B9, but we are not able to have a high taxonomic resolution, whereas before the fewer groups that we get, we're able to have a better taxonomic resolution. And we had predator swamping in both markers. So moving forward, we decided to remove the noise of all these data by removing the Salp DNA sequence numbers or the Salp sequence numbers and other unlikely prey groups that we thought that probably were just maybe part of the net, not really, or the Salps are not actively eating, like siphonophores and gastropods. So we were left with what we found in the literature that has been present in the gut context, which is diatoms, Rhizaria, or radiolarians, syndignales, dinoflagellates, and copepods. So those are going to be from now on the small prey, and all the analysis I'm going to present are based on these small prey groups, those 5 prey groups. So we use the we continue to use the sequence numbers log transform for these prey groups that I mentioned, and we ran a similarity Bray-Curtis analysis, and we constructed the cluster diagrams with those similarity values. Here I'm showing again 2019 on the top and 2019 on the bottom, and in red it's Yasis sonaria, in green Salpa aspera and Salpa fusiformis. So for B4, we see that Salpa aspera and Yasis sonaria kind of clustered together in some cases for both years. So we can see that over here we have one major cluster where there's Yasis sonaria and Salpa aspera, and then the separate cluster for both Salpa aspera and Salpa fusiformis. In a similar case for 2019, where Salpa aspera was mixing with Yasis sonaria, and also Salpa aspera was mixing with Salpa fusiformis, and Yasis sonaria was over here by itself. And now for B9, it was a similar situation where Salpa aspera and Yasis sonaria, the prey similarity was mixing, in the cluster formation, and Salpa aspera and Salpa fusiformis for both years. However, something to note for both markers, for B9 and B4, Salpa fusiformis was only mixing with Salpa aspera. It never mixed with Yasis sonaria. So I thought that was kind of interesting that only those Salpa aspera and Salpa fusiformis were mixed, but not with Yasis sonaria. So from then on, we wanted to find out a little bit more about the diversity of the prey groups. So for that, we used the Shannon diversity index, and I'm presenting the plots of these index values for Yasis sonaria in red, Salpa aspera in green, and Salpa fusiformis in blue. I continue to use the same values for these statistical analysis sequence numbers that were log transformed. And the results for B4 show a wide range of diversity values for prey groups in gut contents of Yasis sonaria. You can see that in 2018 over here in the red, in 2018 and mostly in 2019, we see this wide range of the diversity values for these species, similarly to Salpa fusiformis, right? But Salpa aspera was just not as widely spread. Yeah, and there were some differences between years. And now for B9, the same analysis shows that there was a narrower range of diversity values for prey groups in the gut contents of Yasis sonaria, but there were no marked differences between the Salpa species, Salpa aspera and Salpa fusiformis between the 2 years. So we thought that was interesting, and we wanted to look closer at those groups that we thought might be important or driving this diversity. So we went in and I focused on the Copepod prey species, but for now on, I'm only going to use B4 because, as I mentioned before, it has a better taxonomic resolution and we can actually detect species. So this time we use the average proportion of the B4 sequence numbers that were log transforms. And we detected this in the Salp gut contents of the 3 Salp species. So I have 2018 on the left side of the bar plot and 2019 on the right side, and then all the species listed in the far right. So the Copepod species composition in the gut was different between the years. So 2018 seems, at first sight, there's less colors. And in 2019, we definitely have more species and it's a little bit more colorful. So Yasis sonaria had the lowest Copepod species diversity in 2018, but we also had lowest numbers, sample numbers for 2018, whereas Salpa aspera had the most diverse Copepod diet in 2019. And you can see it was kind of nice to see Copepod species that we were expecting, but they were pretty different between the years. Like Calenus finmarchicus was very predominant in 2019, and in 2018, only Salpa aspera show a couple sequence numbers from them. So again, I think, at least for me, it was interesting to learn more about the diet in terms of the Copepod species that were there. Then we move on to the phytoplankton groups, and we started with dinoflagellates because we saw that when we were looking, when I showed the first plots of the prey groups, dinoflagellates were kind of the most abundant, and there's been publications that report them in the gut content of Salps. So I continue to use the average proportions of sequence numbers, and the Salp gut contents collected in 2019, again, as well as for copepods, Dinoflagellate showed a wide range of species detected in 2019. And again, some species were like Heterocaspaniae, whereas more prevalent in 2019 and 2018. So all of this was pretty cool to see the differences in years. And then finally, another phytoplankton group that we think is pretty important or may play a role in the diet of salps is the diatoms. And in this case, the Salp gut contents collected in 2019, again, had a little bit of more diversity or a diverse diet of diatom species detected in 2019. And more so for Yacisonaria and Salpa aspera. So with all of these results, metabarcoding of Salp gut contents in 18S for B9 and B4 showed differences in the composition of the gut content between the three species, and also the Salp species showed different taxonomic composition of their gut contents between 2018 and 2019. The analysis of the Bray similarity showed clear distinction between Salpa fusiformis and Yacisonaria gut contents, and 18ASB4 taxonomic resolution allowed us to have a more detailed analysis of prey groups in the Salp gut contents. And thanks to that, we observed that dinoflagellates, diatoms, and copepods were more diverse among all Salp species collected in 2019. For future research goals, we already have some larger sample sets coming up, I think 100 and maybe 300. And that hopefully will allow comparisons for the target Salp species between seasons and environments. And also we're hoping to integrate gut content isotope analysis to this data set. Also, in order to consider prey selectivity of Salps, we are hoping to compare gut content data with ambient phytoplankton data. And all of this, it's gonna hopefully lead to more coordination with other ocean twilight zone projects, including the metabarcoding analysis of phytoplankton composition from the water column and the comparison with metabarcoding of zooplankton samples. And with that, I just would like to acknowledge this is, again, research is part of the Woods Hole Oceanographic Institution Ocean Twilight Zone project, funded as part of the Audacious Project housed at TED. And we appreciate the contributions of Paul Kager from Woods Hole and Bo Reese from the University of Connecticut. And this work had additional support by the Department of Marine Science, UConn. So with that, I finish and I can take questions now. I can remember the format or at the end.
Thank you. That was awesome. I liked it. I mean, that's all I can say about it. That was great.
Yeah.
Thank you. Anybody questions?
It was pretty quick, so if you guys
want
me to go back, I can go
back. That was good. Looking forward to hear it again on OSM, I guess. Just squeezing it into 5 minutes will be interesting, but
yeah, I know. I'll just say something about the results. They're very interesting.
That's fine. That's good. Yeah. Any questions?
I have one question for you. I'm not a geneticist, but I'm thinking about maybe doing something with eDNA, but I wondered how, did you have any trouble with the amount of information on the library you used? I missed it on your slide, but yeah, just for sequencing the different species.
Yeah, you mean like how to handle all the sequences, like the amount of data, or more of like detecting the species?
Just in terms of, was there enough information to find those sequences in?
Yes. So the SILVA database has like a wide range. So you can detect from bacteria to like a little bit protozoa, and as you saw, some fish too, right? Like some salps, some fish. But recently there was another collaborator, Leocadio Blanco. He published actually a database that is more curated towards zooplankton, but I think it's more in the water column. So we use that modified version of Silva because we were able to get more groups. However, there's an effort for CO1. I think they're developing like CO1 databases. There are actually even regionals, like you can choose, what region you want to focus your search on. So yeah, if you feel free to like email me. I work with Anne Buckland, and she's part of a group, ISIS group that works on CO1 database development. So depending on what zooplankton groups you're looking to work with, there might be a choice later on about the marker just based on the kind of data you're looking for or the taxonomic groups. So we'll be happy to help you with that if you have any questions later.
Okay, great. Thank you so much.
Yeah.
From the voyage, you collected the samples. Do you also have zooplankton counts to compare what you find in the guts kind of as genetic markers and also what you find in the water column?
I'm not sure about zooplankton counts, but there's another colleagues that are doing the metabarcoding of the zooplankton that was collected in the net. And I think somebody else is also doing eDNA. So we're hoping that, that can eventually make sense a little bit more, like how to integrate all of that information into, Bray selectivity still kind of seems challenging because salps are just filtering whatever it's in the water, right? So, but it's
that's what you think, at least, but of course, it's really the case. Yeah. Oh, interesting.
Right. Yeah. But it's encouraging to see other works. I think there was a recent work, but I can't remember the author, but it was more about, besides, there could be some taxonomic selectivity. So that salps are actually selecting for certain taxa. So I think it's encouraging to see that maybe it's not all about like a vacuum cleaner, right? That they actually are preferring certain things. So we're hoping to see that for the different species. But yeah.
There was another question by Bowen. I was wondering what's the factors that makes a difference of dye composition for Salp in 2018 and 2019?
Yeah, I don't know. It's actually, so 2018 happened around August, and 2019 was July, August. So it's actually pretty close in time. But we were hoping that eventually we will look at environmental data too, to see if that has something to do, maybe the conditions in the water column were different between the years. Who knows, like a warm ring or something like that, or the phytoplankton curve was different for both years. So we're hoping to do that. How do salps move in the water column? Okay. Right. Yeah. So right now I'm trying to wrap up this work, and somebody asked about. Sorry, I'll repeat it. How do salps move in the water column? That could explain the differences. Do they change the diet with size? So yeah, so now that I'm writing more of the discussion area of these results, I'm looking at the physiology of their swim, like the speed, who swims faster, what does that relate to, like filtration rates? Does that mean that, again, like, are they going to encounter certain cells more because they migrate more, depending on the species or the size? So yeah, now we're getting to that part of like trying to like figure it all out, not arm waving, but kind of like putting it all together. So
I think you could do like a salp race. So you put different species next to each other, and then just betting on the salps and who's winning. Anyway.
Hey, yeah, put different foods, see who goes faster.
Yeah, why not?
Like donuts.
Yeah.
Yeah.
All right.
No,
Oh,
yeah,
thank
that's
you
a
so
great
much.
question. Yeah, of course.
That was great. I think we move on now